So this talk was given actually at a public speech in front of, I guess, about 100 or so people, the general public in Philadelphia 4 years ago. And the reason I bring that up is there's some pictures in this talk that are eerily relevant to our current situation involving the pandemic and involving Emory that at the time when I gave this talk were not even on my radar. So I think it's interesting looking back and seeing that's still quite present. And to kind of walk you through the theme of the talk, I'm going to cover about 6 or so main topics. I'm going to keep this big picture, so for those of you that are not scientists, don't worry. Hopefully this will be interesting to you and engaging. For those of you that are scientists, there'll be some science, I promise. I'm trying to walk the middle of details and big picture. So we'll start talking about just how do we kill bacteria right now? Are they overused? And the short answer is yes. Do they have side effects? What is resistance? It's actually something that I think the general public doesn't really have a good handle on what it actually is, and especially given all of the focus on COVID currently, I think there's lots of pressures on resistance currently. And then we'll end on an uplifting note on is the end of the world, are we doomed? So first I want to kind of give a perspective, because as somebody who works in the field of antibiotics and bacteria, I don't think I had a full appreciation of how prevalent they are in everyday life. So to kind of, I like using analogies. I think they're a good way to communicate science and a good way for people to remember things. So bacteria started, estimated, about 3.7 billion years ago. We started about, they're saying about 100,000 or so years ago. So if you put that on a clock, we're basically the last 2 seconds of a full day compared to how long bacteria have been here. So they've had quite a long time to evolve and to kind of develop their niches on the planet. This is something that I think just continues to boggle my mind. If you count estimates of how many bacterial cells are on the planet, it's 5 times 10 to the 30th. So to kind of give you an idea of what that actually means, if each bacterial cell was the diameter of a penny, and obviously they're much smaller, but just sort of for frame reference, and you stacked, you'd have a line that's 1 trillion light years long, which I just think is this mind-blowing number of how big things are and how many bacterial cells are on the planet. If you were to weigh all of them, it's the weight of the entire UK. To give you, again, perspective of the mass. If you were to actually think of your own body, it's about 4 pounds of your biomass is bacteria. Again, it's kind of a creepy kind of thought, but that's how much of your body is actually bacterial matter. And they can live everywhere. So if you've been to Yellowstone, you might have seen these really beautiful hot springs with different colors. Those are all bacterial biofilms that can grow at very high temperature. They can grow. There's been some natural products isolated from bacteria in Antarctica. And they can grow in your bathroom. You search a clean bath on Google. And the reason I bring this up is I'm sure a lot of you are probably familiar with that pink ring or yellow ring that you might see around your sink, around your toilet. That's this bacteria called Serratia morensins, and that's a bacterial biofilm that's formed. Luckily, those bacteria don't really cause any direct harm, but they do cause the transfer of resistance, which we'll talk about later. And it's a major issue now as more of us use Lysol to do everything going forward. So like I said, they're everywhere, including our body. Estimates between 50 and 70% of the cell matter in your body is bacterial, which I think is again, crazy to think half or more than half of your body is actually bacterial, not mammalian. One of my favorite pictures that I give in my talks, this is a used toothbrush bristle. And that, if I can use my mouse, so that is actually Strep mutans, the bacteria that causes decay and caries, also can cause heart disease. And what happens is that plaque that you feel on your teeth, that's actually a bacterial biofilm mass of bacterial matter. And you can have good bacteria. So if you're lucky to come from parents that never have had cavities or teeth issues, you probably haven't either. But if you happen to be like me and come from parents that have cavities, the bad bacteria transfer to you when you're young, and they kind of stay there for a long, long time. And no matter how much you brush, how well you keep track of your mouth, you can keep it at bay, but you're still susceptible to getting viruses or getting infections and cavities. And what's interesting is the way these bacteria work is they take the sugar and turn it really acidic and outcompete the other bacteria. And it's that acid, kind of like if you drink a lot of soda, that decays your teeth and makes it vulnerable to decay. And as a lot of you probably heard, also the gut microbiome is a really popular area of research. And this is something that I'll really focus on for the middle part of my talk. One thing to think about is when we do testing for drugs and approval of drugs, we test them in animals and in humans, but we don't really look at how the bacteria in humans affect those drugs. So one, I think, interesting study done by a former colleague of mine, Emily Valskis at Harvard, they studied how different drugs that were shown to be inactive in some humans, why they were inactive. And what they showed for this drug, digoxin, which is a cardiac drug, is that if you're unlucky to have this bacteria elenta, what happens is the bacteria will break down the compound, hydrogenate that double bond, and make it inactive so it doesn't work anymore. So some things that our lab starts thinking about, and we'll talk more about this at the end, is if you can develop drugs against these certain bacteria, you might be able to gain broader usage of these known compounds that might be inactive in some people because of these bacteria, which I think could be cool. In 2003, Graham Rook proposed this idea of this whole old friends hypothesis, which basically says we've evolved our body to be reliant on our microbiome. We haven't taken, or we have not evolved over 100,000 years with antibiotics, right? That's something that's more recent, the last 100 years. How is that affecting our evolution? How does that affect our tolerance of lifestyles? Is that a reason why allergies are increasing? Is that a reason why celiac disease, all those other things are increasing? And possibly, and this is what a lot of research is going on right now. So I want to talk about how these compounds work and how they're used as a way to kind of give a bigger picture of what's going on. So I think many of you are probably familiar with this story, but back in 1850, about a quarter of human population died before age 1, mostly because of infections. Today, that number is cut down to 0.6%, which I think is really quite remarkable. What Sir Alexander Fleming found in 1928, in this, I think, very famous experiment, was that he was studying how bacteria grew. This is a plate of Staph aureus growing on a lawn of agar. And what he realized after, as he tells it, a long vacation, he came home or back to his lab to see that he had this fungus, so that's bacteria, Staph aureus, and he found this mold, penicillium, bread mold growing on his plate, and you can see kind of that the bacteria died. And this was his first hint that mold was actually producing a compound that kills bacteria. Fast forward a few years, and we now all know that this is penicillin. This is I would say one of the wonder drugs that many say helped us win some world wars and saved our culture, really. What was interesting is this mold, when he isolated it, only made about 4 units, so it would help 4 people per mil of solution. What's amazing is what we've been able to do as in the scientific field is increase it 250 or so units per mil to now. We've engineered bacteria to make over 500,000 units per mil today. And what we do as chemists, and others have done in the field in pharmaceutical companies, is taken penicillin and derivatized it through chemical means to make different analogs, which is great because you can make new generations. The problem is bacteria are smarter than that, and they can evolve resistance faster and faster. So this is why we need to find new actual compounds, not just kind of repurpose old ones going forward. And again, 1945, he and others were recognized for both the isolation and the repurposing and engineering of mass production of penicillin for broad use globally. So now some of these names I'm sure many of you are familiar with, whether for your own usage or through your children. There's some that have been combination therapies given to overcome this penicillin resistance. That was one of the first resistance issues that have come up. But as you can see, a number of molecules out there, mostly back, I would say, in the golden age, from the 40s to the 70s. Not as many most recently, as many have heard. There's been quite a few stresses on antibiotic development currently ongoing. But what I think is interesting is all these compounds are made to help humans, yet the largest consumer of antibiotics are actually farm animals, which this is something that I guess I was a little naive with when I started my career, but as I read more about the use of antibiotics and resistance, this is something that really has intrigued me in terms of the research that's gone on, looking at why this has been done, why animals are being fed antibiotics, and what's the ramifications of that going forward. So why do we give our livestock antibiotics? I think if any of you have heard Mary speak, and if you haven't, she's a great person to have in this lecture. Her and I have talked quite a bit. I teach a freshman seminar on this topic as well, and she's come and speak to my class about this topic, so it's really great to hear the history. But in the mid-1940s, there was lots of competition to figure out what's the cheapest way to have the highest yielding chickens and how much meat they can produce. And what they found, I think this was at Illinois, what they found was that if they fed waste product from the production of penicillin and other antibiotics that had low doses of antibiotics left in that feed, you actually got fat chickens. And they tracked this up to they tracked it down to the actual antibiotic being the reason that you get the fat chicken. It wasn't the feed, it wasn't anything special in the supernatant, it was actually the antibiotic that did this. So people started getting curious and wondered why that was the case. So what studies started showing in the 1960s is if you had a chicken that had a normal microbiome, a normal bacteria in their gut, or if you had a chicken that was born in a germ-free environment in an egg, if you gave one pellet penicillin, you got a fat chicken. If you gave the other penicillin, the chicken stayed the same. So they started tying this idea that it's actually the microbiome of the chicken and how the antibiotics affect the microbiome that's affecting the chicken's growth. And again, going back to that old corollary, we are, basically bacteria. Our cells have evolved to work with bacteria. They work in tandem. When you perturb that equilibrium, other things happen. So it seemed at this point, this is again 60 years ago or so now, that the microbial community played a role in obesity. And this spurred some new research, I'd say, in the last 20 years about looking at this and tying it to humans as well. So something that, I mentioned, maybe I didn't mention this to everybody earlier, I am a parent of 2 young kids. I have a 3-year-old and a 9-year-old. So this is something that really hits home for us as well. I'll get a little more into the details later on, but to cut to the chase, my oldest son has allergies, was born with allergies, and my wife was on antibiotics. My youngest son is 3, was not, my wife was not prescribed antibiotics. He has not had any allergies. Again, small data set. Scientists should use bigger data sets than their own children. I understand. But this got me really interested in this idea of how antibiotic usage preterm affected usage of allergies growing up. So the reason I say that is the top 6 prescriptions for children in 2010 are here. Over 40 million doses for US children. And typically children that get put on antibiotics get put on it for multiple courses when they're younger. And that's really going to be the key to this story as we go forward towards antibiotic usage. So again, why are so many prescribed, and does it matter? So if you're like me and you've had children in your life and they were young, you have a 1-year-old that's got a runny nose, a sore throat, you might have to go in the next day and teach class. So you're kind of really eager to get to the doctor, get them fixed, so they go back to daycare. Typically, you assume that this causes a bacterial infection. If that's the case, antibiotics, great choice. They should work. However, if it's a viral infection, and this is kind of what I hinted at earlier in my talk, this talk was made 4 years ago, and I think a lot of people now recognize what that virus looks like. It's a coronavirus, which I think is kind of fitting, giving our now. Typically, you have a viral infection, and viral infections, hopefully everyone here knows, are immune to antibiotics. It does not affect viruses. Over 80% of these runny nose, sore throat instances with children are viral infections. And again, antibiotics have no effect on viral infections. But I would argue most parents, when they go to the doctor, pay their copay, wait in line for an hour to get to see the doctor, they want something that's going to make them better. And in most cases, almost all cases in fact, if you give antibiotics to all the kids, they will all get better. If it's viral, it'll get better after 3 days, not because of the antibiotics, just because it fights the viral infection. If it's bacterial, it'll be better in 3 days, because that's when the prescription typically runs its course and you finish it over 7 to 12 days. So in all cases, giving antibiotics seems to be, the cure-all. There's no side effects. Typically, there's no issues, right? Everything's perfect. But as we learn more about what's happening with overusage of antibiotics, this is a major issue. And I think a lot of doctors are getting more sophisticated about not overprescribing antibiotics. And to put this in perspective, I think this is crazy. For a child under 2, typically, your child will receive 1 to 1.5 courses on average going in their lifetime. And the average child receives 17 courses before 20, which again, I just think is crazily large for how often they would actually be effective and how well your body can actually affect and fight off these types of infections or viruses. In addition, lots of preventative medicine now take advantage of antibiotics effectiveness. If you go in for oral surgery to remove a wisdom teeth, to get any kind of a tooth replacement, you're typically prescribed premeditatively antibiotics for the risk that you could get strep mutans, that bacteria I talked about earlier, into your gums and into your bloodstream and cause issues going down the line. They don't see an actual issue with giving you antibiotics. So they give it to you preemptively ahead of time, prophylactically. Something I'm going to talk a lot more about, if you're pregnant, you get tested in your last 2 weeks of pregnancy. If you test positive for group B strep, you're prescribed prophylactically antibiotics, because group B strep has been shown in cases up to 1% of newborns are stillbirth because of infection of group B strep. So now, again, 25% of women are now taking antibiotics before they give birth. This is up from, I think it was about, so it's 40% versus 25% 20 years later, so it's doubled. And again, it's true, a 1% chance of your child not, of a stillbirth is terrifying. And this isn't saying we shouldn't prescribe antibiotics. The way I'm looking at this is, if I can develop an antibiotic that's specific for group B strep, which is the issue, I can have a new prophylactic treatment that's prescribed to all women that are pregnant and limit the collateral damage to their microbiomes. And you'll see what that happens later on in the talk. So overall, in 2010, and a little bit dated, but 10 years ago, over 260 million courses of antibiotics in the US, almost one a person, which again, I think is quite a large number. And because of this, we have these unintended consequences. So this is getting back to, historically, we always hear about antibiotics being terrific. They really have very minimal side effects. To me, they really are these wonder drugs. They target things that are only in bacteria, typically, and that shouldn't have an effect on humans. And we take large doses, these big pills, if you've ever had them, that are sometimes quite uncomfortable to take because you need these large doses to take out the bacteria. But there are these unintended side effects that you might not be familiar with. So a book that I highly recommend for book clubs, for people that are interested in this topic more, is this book called The Missing Microbes, written by Marty Blaser. So Marty used to be a faculty at NYU. He's since moved to Rutgers. Incredibly generous colleague. We've had discussions about these topics in the past. He's just, I think, a fantastic scientist doing public health research. And what he is focused on his career is looking at how antibiotics affect the microbiome and correlation to things that we've mentioned: obesity, asthma, allergies, upset stomach, things like that. And you might ask, how do you study these types of things? And there's two ways that we're going to talk about today how you study them. The first is looking for correlation in these large longitudinal studies that are, I think, very well known to people that do research in public health, in which you look at personal studies over time, surveys over time, and how you can analyze them in ways to get to what you're trying to study. Have lots of different preemptive rules that are in place. So we'll talk about that part first. And one study that he did that I think is really, I thought really, for me, eye-opening, and this was about the time my first son was born, is he looked at the differences in childbirth between a traditional childbirth with no antibiotics, one where the mother took antibiotics beforehand, and a cesarean C-section. And what he discovered is, or kind of walking you through these and why he was interested, if you have natural childbirth antibiotic, you pass your microbiome of the mother directly to the child through the vaginal canal. If you have an antibiotic, now it's a new culture, a new microbiome that's being passed because the mother has been exposed to this antibiotic, and it's pretty much non-discriminately killed lots of things. So the microbiome is now selected for arguably the more pathogenic and the more harsher condition living bacteria. And in cesarean child, you actually have a whole different bacterial colony passed, typically that of the sterile, hopefully sterile, hospital room, and whatever they acquire early in birth. And what he was curious is if he could divide into these three subgroups, what would he find? What he found typically is lactobacilli are the good guys. This is what you get actually in these probiotic yogurts and things like that you take. Those are what really helps survive. And long story short, if you lose them, it starts getting harder to have this equilibrium in your body. One of the studies he looked at was 15,000 about pregnant women in 1991. And what he saw is that 33%, on par with what we said earlier, received antibiotics. Of that group, 22% of the children are likely to be overweight at age 4. 9% were born via cesarean C-section, and now 200% were more likely to be overweight at age 11, which is crazy to think about this. Again, it's a moderately small data set, but these are real, I think, real things. And if you think about our society as a whole, you don't really hear much about part of our obesity culture. You hear about soft drinks and otherwise, but it might be more than that. Actual body being able to break down what they're eating and having different effects. A second study I think is even more telling. So this was over 600,000 people they followed in Denmark, looking at Danish children, and they followed them pretty regularly over 6 years. I guess over 8 years, sorry, that's a typo. 85% received antibiotics over that course. So this is now we're looking at the children from birth, from birth to age 6. The study was over 8 years. From birth to age 6, 85% received antibiotics over that time frame. In that group who received antibiotics, 84% more likely to develop inflammatory bowel disease, again, which makes sense if that's where most of your bacteria are. 300% more likely Crohn's disease, again, 7 times the risk if you had more than 7 courses. And I talked with Marty about this. Again, this was right after my son Max was born. He said, be really careful getting antibiotics, especially in your first 6 months. That's the key time to build up your microbiome. There's still time to transfer your microbiome from the parents to the children through kissing, interaction, otherwise. But after that 6 months, it kind of gets established, and it's hard to change and perturb that environment. And again, is this just for infants and toddlers? No, I mean, I don't enjoy doing this, but if I hear somebody has a gluten allergy, I typically ask them, have you had antibiotics? And it's surprising how many times people said, oh yeah, I actually did just take antibiotics recently. And since that time, it's flared up their gluten allergies. There's been research at another school, McMaster University, that's shown certain bacteria break down gluten ways that then your body recognizes as a threat, and it generates an immune response, and it heightens this gluten allergy significantly. So again, if we can figure out what bacteria are doing this, we can possibly have these narrow spectrum targets that target these bacteria and help with these situations. And again, on average, they had half the amount of certain commensal bacteria that we termed as the good guys. And I would say in the last 5 years, there's been an incredible amount of research toward what's the ideal microbiome. There's a former colleague of mine who's now a professor at Stanford, Michael Fischbach, is looking at, can you create a bacteria that best replicates the human body, and then start picking out the good guys and the bad guys, and coming up with, this term, I don't really love the term, but this term, the poop pill, what kind of microbiome can you re-implement into people, and what bacteria should be in that pill to help repopulate the gut to make it more beneficial going forward. I should also mention there's actually been a lot of significant ties recently. You might have heard about this compound, colobactin, that's been shown to be tied from bacteria to colon cancer and other types of cancer in the intestines. Basically, this bacteria can produce a toxin that triggers a response on your DNA that causes cancer as well. So there's also other types of cause and effects, not just allergies, gluten. There's actually ties to cancer and other things as well. But I should be clear, there's lots of public health studies out there. You'll read many that say coffee is the worst thing you can drink, coffee is the best thing you can drink, soda, alcohol, all these things. Correlation doesn't mean causation. It really, the studies that then go back and test these in animals, I think really are the ones that have the most robust data. So what Marty's been doing recently in his lab is taking mouse models, similar to those chicken models I mentioned earlier, and studying how populating them with different bacteria affects their growth. So one of my favorite experiments he's done is he's looked at a normal mouse and genetically identified the bacteria in that mouse, treated it with penicillin or amoxicillin, and then looked at those new populations and identified them through DNA sequencing what they are. And then he takes germ-free mice. These are mice you buy from birth in a bubble. They've never been exposed to any bacteria. They're totally innate to seeing any kind of bacteria. And he populates them directly with the bacteria from the gut of a normal mouse. What he sees is that mouse comes out normal. But if you populate now with the bacteria, so this is the opposite of the experiment I said earlier, just the bacteria of the mouse treated after penicillin, so no penicillins involved, just the bacteria itself, he actually shows you get fatter mice. 10% more weight and 40% more fat cells. So the metabolism actually is changing of the mice, which again, if you think about how bacteria work, how your body works, this makes a lot of sense. I think a lot of times I've always was told when I was younger, on the thinner side, that I have a really active metabolism, and your metabolism changes over age. The more I've researched, the more I've read, the more I realize that's probably my bacteria in my body that caused that. And you might have certain bacterial genes that cause metabolism to change. As you get older, that might change also from what antibiotics you've been on, your environment, where you live, the temperature, all these things play a role in the hospitability of your body for bacteria. And I think a lot will be learned in the next 20 years about how this fluctuation changes how your body addresses stresses. And again, as I mentioned earlier, allergies and obesity are bad. We can live with that. Resistance, I think, is actually the bigger issue, and I see this being actually a large problem in the next 10 years. So I'm going to switch gears now. That's the part actually of my talk I like giving when I give my research talk, because I think a lot of people don't understand the adverse effects of bacteria in the commensal microbiome. I'll talk to almost anybody and they know antibiotic resistance is an issue to certain extents and how it is. One thing I thought was really interesting in my public engagement work is I was reading through work from the World Health Organization, other, I think, fantastic communicators about antibiotic usage. And over half the population actually doesn't know what antibiotic resistance means. So most people, and hopefully no one in the audience, but if you do, hopefully I can educate you on this as well. Most people think that your body becomes resistant to the antibiotic. So if you have antibiotic resistant infection to penicillin, they think your body's resistant to the penicillin and you can never take that drug again because it won't work because of your body, not because of the bacteria. What it actually means is the bacteria are now not susceptible to the antibiotic and that they become resistant, not your body. So this is something that to me was always obvious, but it's something that I never thought deeply about, the word use, the word choice, and how we talk about this. So this is why we talk about when we use the term antimicrobial resistant, or it's antibiotic microbe resistance, and it's putting the microbe and the bacteria in the terminology, not just resistance to the antibiotic, to tie it all together. And you've also might have heard of this term superbug, all these other issues about the bad bugs out there. I think this graphic really sums it up nicely. What are the actual superbugs that you've probably heard about around here? We're lucky to be by the CDC, so you actually hear a lot about what they actually are. Strep pyogenes is this bacteria that causes strep throat, sore throat. It's bacterial. MRSA is probably the most common bacteria everyone's heard of. Gonorrhea is also a bacterial infection. C. diff is the one that you've started hearing more about in terms of irritable bowel syndrome and gut issues with excess antibiotic usage. E. coli is typically the bacteria you see if you have food poisoning from different types of lettuce or things like that in the news. And Acinetobacter baumannii is a newer pathogen of interest. It's typically in the veterans hospitals. It's been brought back from the Middle East. And it's actually a really nasty pathogen. And we're lucky at Emory to have really, I would argue, some of the best facilities to do this research. I was a little naive at the time when I moved down here, and I was fortunate to join the Antibiotic Resistance Center we have at Emory, and I'd argue we're one of the top centers for antibiotic development in the country. And I think David Wuest does a fantastic job running that institute, and I wish the university itself would actually advocate this a lot more to the general public. I think we have a lot of really great resources available for these studies. But again, how do these superbugs develop? Where do they come from? Where do they exist? So this is the part of my talk where I will start delving more into my research. And what I always like to disclose about this part is this is the research I don't love talking about because it's so simple. And I've come to realize as a scientific academician, the simple stuff is actually the stuff that's the best to talk about because people can relate to it, engage in it, and it actually is the most relevant to others in terms of where the money is, to be honest. So our group has had an interest in disinfectants. This one I show here, you won't see anymore because it contains triclosan, and triclosan has now been banned from all hand soaps, which I think has been a great move. A lot of companies thought by putting in an antibiotic, triclosan into everything, it would make it better. It's actually made things a lot worse. And it's made resistance a major issue. And to be honest, it's overkill. Something that the last 6 months has taught me is that you would never talk about something to the media. I never knew how important it would be to talk about hand washing and soap. For those that haven't seen, I've been caught in the news a couple times on the Weather Channel, CNN, and otherwise talking about hand washing and soap. I never thought as a scientist I would talk about these principles. It's actually been fantastic because I was able to use some of my material that I teach to freshmen in Chem 202 in our new curriculum on TV, talking about entropy and how washing your hands and soap ties all these things into effect. I think it's really been awesome and fun to do. But you don't have to worry about antibiotics. Hand soap works just fine. Just use regular bar soap and you'll be fine for COVID, for bacteria, all those things. What everyone now is interested in, and this is where our research really I think is exciting, is in Lysol. So Lysol are these compounds. They're called quaternary ammonium compounds. They've caught the news quite a bit. I've also been talking to news agencies. I think I had a Washington Post article out recently on different cleaning agents along with Cassie Quave at Emory, on bleach, on natural products from, we've heard our president talk about oleander and other things. And these are called quaternary ammonium compounds. They've been around for 100 years, and I would argue they're probably the most effective cleaners we have. These molecules, the one on top is the active ingredient in Lysol. It's called Lysol because it lyses bacterial cells. Those positive nitrogens bind to the negative lipids on bacterial cells, on coronavirus, to your actual mammalian cells as well. And that little tail in the end wedges in like a sword and breaks open the virus, breaks open the bacteria, and works incredibly effectively. What a lot of companies have now done is they've taken out triclosan and they've put it in benzalkonium chloride, BAC, into the hand sanitizers and made them antibacterial as well. I've gotten lots of questions about, do you have to buy Lysol to clean your house for COVID? If all you have to do is look at the generic brand, if you see benzalkonium chloride, anything with that word alkonium on there, it will be effective. It will work against coronavirus. So don't worry about having to find Lysol or Clorox. You can find others with also, about 60% alcohol. All those will be effective against coronavirus. And what's crazy is these cloths are used everywhere. And I would argue that they're the most common disinfectant that we have out there. So in 2016, when I gave this talk originally, this was a week before it, there was an article that came out in the Wall Street Journal that said, are antibacterial soap safe? I think it was 2 months later, the FDA banned triclosan, but they still continue to allow benzalkonium chloride to be put in soaps. So if you see an antibacterial soap by label, look at the back, you'll see that compound. What I found ridiculous is this is research I've done in collaboration with Villanova University. We've published over 20 papers with undergraduate authors at Villanova with one of my friends who's a professor there in the chair of the department. I never thought this research would take a twist into resistance. I never thought bacteria would be able to resist lysing their membranes or their cells. This all came up through the company that Gretchen mentioned earlier, Novalice, that we developed at Temple. that was on these new compounds that we developed to overcome resistance that actually is a major issue. So what everyone does, they take their Great Value or their Lysol spray, spray it on your countertops. One fun fact, don't wipe your countertops after you apply Lysol. It's got to stay there for 4 to 5 minutes. Something, again, I didn't know. This is why they typically look wet, let it evaporate. Once it evaporates, you're fine to clean it off. Typically, the chemical that you're using on the counter actually is fine and safe. You're not going to consume it at any level that's going to be toxic. So I wouldn't worry about it. But if you want, 10 minutes later, wipe it down with a sponge, you'll be fine. After you spray down Lysol, you will have some bacteria that's left over. It kills 99.99% of bacteria. 0.001 is still, I think it's usually 2 or 3 log of bacteria is still there, still a small amount. And these are now getting pre-selected for resistance. What a lot of people don't realize is if you think about earlier in my talk with those pink, bacterial rims around your sinks or your toilets, you're cleaning them with Lysol, right? You're wiping them, and then what do? You turn on the faucet, runs down, dilutes that chemical, goes into the sink, into the water, into the sewer. Now you have low concentrations of a disinfectant. Bacterial resistance is happening constantly, over and over and over decades. It worries me a lot to know right now that everyone in the world is using Lysol at ridiculous amounts. And we've shown in our lab that you can form resistance to Lysol and actually certain compounds even faster. This is going to be a problem. The good news is we have a solution to the problem, but nobody wants to really pay us for it yet, but maybe soon. I have a conflict, so I will disclose that conflict. I have, again, this technology on these new compounds, but I do think this will be a major issue going forward. The reason this is going to be an issue is certain bacteria, like the ones that make that pink rim, are really good at developing resistance and then trading it to their buddies in the sewer systems, in the soil. They use what's called horizontal gene transfer, and they transfer, like baseball cards, these resistance genes that the way bacteria resist Lysol is they change the membrane slightly and they put efflux pumps in to get rid of it as quickly as possible so it can't build up on the charged surface. It's similar to how the drug colistin works, if you're familiar with colistin. What we showed in an earlier study, and now this is getting more and more dramatic. Again, I wouldn't say this is our best representative trend line, but earlier, no one was aware of what's called quat resistance, quaternary ammonium compound resistance. In hospitals in the last 10 years, they found up to 90% of bacteria in hospitals still have this resistance gene. Again, I'm not trying to fear monger. It's not at a huge state yet. But when you have these biofilm nooks and crannies with these resistance genes, it gets to be enough where it could be an issue. And I would be willing to bet a lot of money in 10 years, it's going to be an issue in hospitals. So it's something that I keep on everybody's radar. Again, bleach, alcohol at high concentration still works well. We still have ways to get around this. But this is right now one of the frontline ways to treat hospitals and clean surfaces. And I think more people in society need to be aware of this. And like I said, MRSA specifically is the ones that's become the most resistant to these quaternary ammonium compounds. So resistance isn't just present to Lysol, it's to all the molecules we talked about earlier. In some cases, resistance happens almost immediately. In some cases, we found resistance before the drug actually is on the market. And the reason being the bacteria that make these antibiotics have to protect themselves from these antibiotics. So they'll make their own ways to defend themselves and then trade those genes to other bacteria to then defend themselves against their competitors. It's really, I think, amazing how this kind of chemical civil war has been going on in soil forever, and you have all these new compounds that are being developed. So is there any hope to this? What's next? Are we doomed? So this is where I think our group comes in, and we look at things from a different perspective. As Gretchen mentioned, I do have a business background. I started a consulting club. I've started companies. The Georgia Research Alliance recruited me here for my commercial interests. And if I were to tell somebody in Silicon Valley about investing in antibiotics, they would laugh at me. It's quite possibly the worst investment opportunity out there. If I make a great drug, it's going to be saved for the worst case situation. It's going to cost me hundreds of millions to develop, and no one's really going to want to use it until the last resort. So we've written some perspectives on ways that the government can help with this. I think it's a little short-sighted. My perspective is, instead of thinking about developing the last resort drug, can we develop narrow spectrum therapeutics that can serve a large population and be used on demand regularly, prophylactically, for women that are group B positive, for oral surgery, but also used for last resort if you have these really bad pathogens that are resistant to everything. So what we've thought of is penicillin is this broad spectrum, kills all the bacteria, including most in your gut. What if we came up with something that was very narrow spectrum, only targeted the bad guys? So this would be narrow spectrum therapy specific, akin to nuclear warhead versus kind of a heat-seeking missile. What if we could come in and hone in on the bad bacteria, Pseudomonas, MRSA, others, they all have their own Achilles heels. Can we identify those Achilles heels and then develop specific drugs that target those specific in ways that are unique and would also give us, some translational opportunities? Sounds great. The issue is how do you find a molecule that's very specific? You can screen, perhaps. You can look in the literature. Perhaps what our group has done is kind of done a combination of both. We look to see where in nature is this happening, and we read through literature and say, is there a compound that looks like it could have these attributes but might have been overlooked because it was a cancer screen or was looking for other attributes for this molecule? How can we find it for bacteria? And really what we've kind of focused on for one aspect of our research is what's called the rhizosphere. So if any of you are gardeners out there, you'll know that the plants, the root system around plants is really rich in bacteria. This is why you compost, you use manure and other things. That bacteria cultivates the root system. And what you have down there is bacteria that want to help the plant grow, and the plant gives its nutrients to grow. There's bacteria that want to kill the plant, and there's bacteria that want to kill the bacteria around the plant. So it's this big kind of, again, little war zone that's going on where it's very close to the root system, and it's developing specific gram-negative antibiotics typically in these environments. And the reason we care about that is one of our specific bacteria of interest in my lab is Pseudomonas aeruginosa. I would say it's most well known for patients with cystic fibrosis. If you have a cystic fibrosis infection and it becomes plagued with Pseudomonas, a lot of times they call it a death sentence because you're not able to be treated with lung transplants and most antibiotics don't work anymore. So if we were able to develop a narrow spectrum Pseudomonas specific therapy, we actually could come up with a compound that would be able to be given to CF patients forever and would have a low risk of hurting the microbiome and keeping them free of Pseudomonas infections. This is also very prone, so if you get a wound infection, a lot of times that wound infection is from Pseudomonas, and it might be out in your garden, you might get pricked with something and you're on some immunotherapy, and a rose bush might give you a Pseudomonas infection or other things out there. A lot of times these bacteria are benign. Your immune system can take care of them, but if you're immunocompromised in any way, if it just happens to be in an area that never heals on its own, this is where these chronic wounds happen that tend to be resistant to most bacteria or most antibiotics. And the main one for cystic fibrosis is a strain of Pseudomonas called PA14, and that's something that we research in our lab. So how do we do this? What do we do as a chemist? And this is more on the research side. So as chemists, I've always been intrigued with the actual small molecules that are in the rhizosphere and the structural pieces of them. So they make these molecules called siderophores. These molecules bind iron. Iron 3 is in low concentration in the soil, and bacteria need it for their oxidative processes, Krebs cycle, and other types of metabolism. So they've come up with these very specific molecules that pull iron out of the soil, and out of humans, in fact, and are able to use it for their own processes. They make molecules called quorum sensing molecules. These are molecules that help bacteria talk to each other. There's been lots of, I think, amazing research on quorum sensing by Peter Greenberg, Bonnie Bassler, and others. Rhamnolipids are compounds that bacteria make that are kind of like the oil slicks of bacteria. They keep bacteria away and you can't get into their area to grow. And of course, they make antibiotics. So what my group does is we go through the literature over the last 100 years and we look at the structures and see if they have any reminiscent structure affiliation with these molecules and try and rediscover or identify molecules that might have been overlooked for their specific activities. And we've gotten quite a few success stories through this. And we think this is really exciting because we've now shown that we can discover narrow spectrum antibiotics by looking at specific mechanisms that are prevalent for certain bacteria over others. And I think this could be the future, as over 90% of antibiotics on the market were derived from natural product structures, whether they're chemically manipulated or used as is, they all come from these complex structures. So now here, I mean, most people in the audience, I assume all people in the audience, do not have access to a chemical hood. You can't do the research anymore. We'll do our best there, but you all can help as well. And this is kind of my public engagement cry to you all, who I think most in the community are actually incredibly active in doing these types of things. But how can you all help? So one, I think most people in this audience are well educated and informed and can make these informed decisions about, maybe passing this information on to your children or your grandchildren, telling them about maybe not getting antibiotics all the time, waiting a couple days before taking the antibiotic dosage, having some patience. Pick the right cleaners, look at the labels. As I've told most people, soap works just fine. You don't need Lysol, you don't need ethanol, you don't need those other special cleaners. Soap works great. If you need to use Lysol, I think it works well. Other tips I've been giving the media: don't pre-make bleach solutions. If you make a bleach solution, try and use it that day. It only lasts about a couple of days. Otherwise, the bleach breaks down and it's not active anymore. Don't make your own cleaners at home. It's a bad idea. I strongly advise against that. Read labels and be conscious of all those things. Spread the word on social media. So I am quite active on social media trying to educate about antibiotic resistance, ways you can help. I think there's lots of benefits, if done right, on Facebook about kind of policing the education out there. There's lots of fear factors that are going on, especially about COVID and otherwise. If you're educated, use your platform in educated ways to share with others that might not be as able to. Research, donations, support. I think donations to universities and foundations are really important, especially now we get squeezed quite a bit. I try and tell general public or advocate for your tax dollars, right? Something people don't realize. There's a 700% return on investment in funding research at universities, which I think for starting companies, Google was founded on an NSF grant, and that's done quite a bit of, arguably good, maybe some bad. But for the most part, lots of job creation, lots of wealth created. Lots of these drugs have been discovered. So get involved in government. The reason I talk about drugs is, again, this was done before I came here. Emtreva, I think, is a great example of this. Lyrica for Northwestern. These are drugs that have made lots of money for the universities, but all were founded on NIH grants. They started with NIH funding, and then were able to be extrapolated out to actually commercially used drugs, which I think really shows the return on investment we get, especially for antibiotics. Pharma companies have all left. I think this is a great area for research. I tell my students, if you get into antibiotic research, you'll never have to worry about a job because resistance is always going to be an issue. It will always be a problem going forward. So being aware of that, I think, is really important. And again, this government funding of basic science, I think, is really critical. Something I'm indebted to is the Georgia Research Alliance. I think they've done a fantastic job funding science in the state of Georgia. If there's some place that you're looking to maybe help support, I know they're always looking for donations. They are fantastic. They try and create jobs in the Atlanta area and the Georgia area, both in agrochemicals, agriculture, science, technology. They supported my endowment. They support faculty at Emory at the medical school. I think they've just been a fantastic player in the state and have also shown multiple X return on investment in their funding. And again, 70% of all funding for this research is from the government, which I think we keep needing to think about that going forward. So with that, I didn't really talk much about my research. This is my team of researchers that I'm totally indebted to. It's a fantastic group of scientists. Marty Blaser, I said, I think also doing some great research. Check out his book. They're pretty active again on social media. We have an active website talking about our recent publications that I didn't spend too much time talking about. Emory and the GRA have been fantastic in supporting us as well.